In prophase of meiosis I, duplicated homologous chromosomes pair and align end to end. Crossover can occur between aligned chromatids, leading to exchange of homologous segments by homologous recombination. Chromosome segregation through meiotic divisions leads to novel genotypes, first in gametes, then in offspring. What's the mechanism of this process of shuffling genetic information? When maternal and paternal chromosomes pair in a nucleus and two aligned chromatids interact, it's the DNA that undergoes recombination to exchange genetic information. In the original Holiday model, single-strand breaks occur at the same point on one strand of each parental DNA. Free ends of each broken strand then migrate across to the other DNA helix, where the invading strands are joined to the free ends they encounter. The resulting crossover junction is called a Holiday junction. As each crossover strand reanneals to its original partner strand, it displaces the original complementary strand ahead of it, causing the Holiday junction to migrate. This creates heteroduplex DNA segments. Cleavage and rejoining to reestablish two separate DNAs occurs in two ways. This is most easily visualized by first rearranging the Holiday structure. DNA molecules with this apparent structure have been observed. This structure can be resolved by cleavage in the horizontal plane, leading to two molecules that do not show crossover of markers in genes A and B. If instead the same structure is cleaved in the vertical plane, both of the resulting recombinant molecules show crossover of markers in genes A and B. All products, regardless of cleavage plane, are heteroduplexes in the region of Holiday junction migration. In a newer and evidence suggests more accurate model for homologous recombination, a double-strand break occurs in one of the paired DNAs, followed by enzymatic trimming back of nucleotides on the new single-strand ends. A free 3' end invades the unbroken helix and displaces a loop of single-strand DNA. A DNA polymerase elongates the free 3' end of the invading strand, further displacing the looped-out strand, which then pairs with an exposed single strand on the opposing helix. The displaced strand serves as a template for enzymatic extension from the 3' end of the paired single strand, which eventually crosses the junction and switches templates. As 3' and 5' ends meet, strands join to form two Holiday junctions. There are two ways to resolve each Holiday junction by single cleavage and rejoining. So there are 4 ways to resolve the double Holiday structure by two cleavages and rejoinings. 2 of these combinations of cleavage and rejoining generate non-crossover recombinants. The other 2 combinations of cleavage and rejoining generate crossover recombinants. All recombinants include heteroduplex segments.